Hello, I'm Sophie Rovner from the American Chemical Society. Welcome to this media briefing from ACS Fall 2022. Lupus is an autoimmune disease that attacks the body's tissues, and it can be fatal. There's no cure, so current treatments aim to limit damage and keep symptoms in check. Some of these therapies have to be injected, while others have serious side effects, and many aren't very effective. But today, scientists report they have begun phase 2 clinical trials with a new type of treatment. This pill contains a compound that, in mice, not only prevents lupus symptoms, but also reverses organ damage caused by the disease. To give us more insight on this work, we're joined by Dr. Alaric Dykman from Bristol Myers Squibb. Thank you for joining us today.
It's my pleasure to join you. Thanks for the invitation.
To get started, can you tell us what problem you were trying to solve with this research?
Sure. As you pointed out, lupus is a devastating disorder, and yet only a limited number of new therapies have been specifically developed for the disease over the past 50 years or more. Some of those new therapies, such as belimumab and anifrolumab, were certainly welcome advances to lupus patients, but they are large molecules which need to be injected rather than taken orally. And they've shown only modest improvement for patients, and they don't work for all patients. Our research was aimed at discovering and developing a targeted therapy for lupus, which could be taken orally and hopefully provide superior efficacy at the same time. We were also aiming for a mechanism that could pair well with the use of steroids, which are commonly needed to treat lupus. We focused on a pair of proteins called TLR7 and TLR8, which were believed to be very important in lupus, and we then sought out a way to prevent them from being activated in patients using a small molecule to block them. It was our hope that if we were successful, we could give lupus patients a better, more convenient way to treat their disease.
How did you study this?
Well, the compound that we're highlighting is called afimatorin, and it ended up being a very effective blocker of TLR7 and TLR8. The discovery of afimatorin was a result of a years-long drug discovery effort that involved numerous teams of researchers here at Bristol Myers Squibb. One key aspect of the process was a screen of our very large compound collection. So after evaluating more than 1 million compounds in our collection, we identified a range of compounds that could block the signaling of TLR7 and 8, at least in cells. This was exciting, but we then had to prove that the compounds were acting in the cells the way that we wanted them to, which was to inhibit because of direct binding to the targets TLR7 and 8, and not by other unexpected ways. So that deconvolution process was quite slow, but eventually we were able to confirm that a few of the hits did indeed function by directly binding to the TLRs of interest and then shutting them down. We got definitive proof of that by generating an X-ray structure that showed our compounds directly bound to TLR8, which was a major milestone for the program. It was really a great day for us when we were able to see that structure and have the confidence that what we thought was happening with our compounds actually was real. So at that point, we had what we called our lead compounds, where they have the starting point of activity against the targets, TLR7 and 8 in this case, but they needed significant optimization. We needed to make them more potent for TLR7 and TLR8, more selective so that they didn't interact with other proteins that we don't want to affect. And then we had to tune the compounds so they had the right properties to be taken orally rather than being injected, and so that we could then study them in animal models of the disease. That process alone took a few years to complete.
So then what did you find out?
The first remarkable finding was the identification of the small molecules themselves, and that they could actually bind directly to TLR7 and 8 and inhibit their function, confirming these as valid drug targets for this modality. At the time we made this discovery, that had not been clearly established in the literature, for small molecules at least, and there were serious concerns about whether this kind of approach would even be able to work at all. Next, we demonstrated that the direct inhibition of these targets by our compounds in animal models of autoimmune disorders did indeed lead to some pretty amazing benefits. Specifically, in mouse models of lupus, we were really impressed with how well afimatorin performed. These models of disease are very challenging, with the mice eventually dying if left untreated due to how severe the disease symptoms become. But we were really excited to see that treatment with our compound could completely prevent the deaths in these models at fairly low doses. We were able to then show that not only could we prevent the development of lupus-like symptoms in these animals that were receiving our compound when it was given before the disease developed, but if we waited until the animals had already shown signs of disease before treating them with afimatorin, we could actually reverse the symptoms of organ damage that had already occurred. Reversal such as this had not been demonstrated with other mechanisms that we'd evaluated in the animal models prior to this work, so we really felt like we were onto something special with this approach.
And I understand that afimatorin also worked well in combination with steroid treatments in mice. So could you tell me some more about that and why that matters?
Yeah, that's correct. Another key finding was that inhibition of TLR7 and TLR8 combined very well with steroid treatments. We first observed this using experiments done in cells, where we tested the ability of our compound to prevent the cells from becoming overly activated. We showed that the combination of afimatorin with a steroid worked much better than either of them alone in preventing that undesired activation. Then we looked in the mouse models of lupus, where we found that combining afimatorin with the steroid prednisolone provided the mice with greater protection from disease than the same dose of either compound given alone. So it was really great to see that the treatments were working well together. This is important because steroid therapy is a mainstay of lupus treatments, but it does come with some safety concerns, especially at the higher doses that can be needed to control more severe forms of lupus. So the ability to reduce the level of steroids in lupus patients or potentially eliminate the steroid use altogether would be a significant advance for any medication. We believe that the notable results with afimatorin in the animal models of lupus on its own and when combined with a steroid really bodes well for evaluation of afimatorin in patients with various forms of lupus.
And what stage are you at with the clinical trials?
Afimatorin has already been evaluated in several phase 1 human clinical trials. Those are trials that involved healthy volunteers and were intended to establish the initial safety of the compound, as well as inform on the human pharmacokinetic or PK properties. We were pleased to find that across a range of single and then repeated dose levels, afimatorin was well tolerated. Furthermore, the human PK properties were quite excellent, providing sustained exposure of the compound through 24 hours with a single oral dose. Measuring the target engagement from human samples then demonstrated that the treatment with afimatorin was able to almost completely block signaling through TLR7 or TLR8 with low doses that were studied. And on the success of the phase 1 trials, afimatorin was advanced to a smaller trial in patients, specifically with cutaneous lupus, as well as to a larger phase 2 trial in patients with systemic lupus, also known as SLE. Phase 2 trials are where the first evidence of efficacy against the disease is able to be clearly defined, so we eagerly await the data that's going to be coming from those ongoing patient trials.
How long might they take?
One of the challenges with developing drugs for lupus is that the clinical trials do take quite a long time compared to trials for some other diseases. In order to get a confident answer that the effectiveness of the therapy, phase 2 trials for SLE can routinely require 3 to 4 years or even longer in some cases to complete. For the afimatorin trial, phase 2 trial, patients will receive the compound or placebo once daily orally for up to 48 weeks with careful evaluations along the way. Getting that completed on more than 300 participants is a substantial effort. So the phase 2 trial began last year, and we expect that the primary portion of the study will complete in the middle of 2025.
Apart from lupus, what other applications might this treatment have?
With the important role that the TLRs play in the activation of the body's immune system, there are a range of autoimmune disorders, aside from lupus, that could really benefit from inhibitors such as afimatorin. Psoriasis, Sjogren's syndrome, and rheumatoid arthritis are just a few examples. Additionally, studies have been published by other groups that link the signaling through TLR7 and 8 to fibrotic disorders such as liver fibrosis, as well as to cardiovascular disorders such as atherosclerosis and heart failure. So inhibition of TLR7/8 could be beneficial to patients in those settings as well, but much more work would need to be done to confirm that.
What other Bristol-Myers Squibb lupus treatments are in clinical trials, and how do they differ from afimatorin?
BMS has had a long-standing interest in developing possible therapies for lupus, having advanced multiple different mechanisms in clinical trials for this disease over the years. Currently, there are 3 different compounds from BMS in phase 2 trials for lupus: deucravacitinib, bronabrutinib, and our afimatorin. Deucravacitinib is the farthest along in the trials, and based on recent positive findings from the phase 2 study with it, it's now advancing into a phase 3 study for SLE, which is a huge milestone and one of the companies very proud of. Like afimatorin, deucravacitinib is an oral medication, not injected. It differs from efimatorin in its mechanism, as it inhibits a kinase called type 2. So it affects pathways that are somewhat distinct from TLR7 and 8, the pathways that are blocked by efimatorin. But I have to say it is really important that numerous approaches are being evaluated clinically for lupus by BMS and by the whole industry, because the way the disease presents in individuals is so diverse, with some groups of patients responding very differently to a given treatment than how other groups of patients will respond. And it's unlikely that any single approach will provide maximal relief for all lupus patients.
So in closing, what do you want viewers to take away from your talk?
I hope that I'll be able to convince the viewers that TLR7 and TLR8 are pathways that are potentially very important to the development and progression of lupus. I'm also looking forward to being able to highlight some of the significant efforts that were required to identify and then to optimize the inhibitors of this pathway and ultimately discover efimatorin. And I hope that I can convey just how committed BMS is to the lupus drug discovery, with efimatorin being representative of that commitment.
Thank you, Al, for sharing your research with us.
It was my pleasure. Thank you so much, Sophie, for giving me this opportunity.
Be sure to check out our other media briefings for ACS Fall 2022, which will be posted throughout the meeting at www.acs.org/ACS Fall 2022 briefings.